Hello and welcome back to A Level Biology Help. Today I'll be taking you through the genetic fingerprinting section for AQA A Level Biology. Near the end of the video I'll be taking you through a few exam style questions and explain their mark schemes. And as always, in the comments there will be timestamps so that you can skip to the different sections of the video if you do not wish to watch the whole thing. Righty ho, let's get started. So what do we mean by genetic fingerprinting? Well, genetic fingerprinting is something that you might have heard of before on TV to investigate crime scenes. So genetic fingerprinting is a method used to produce a specific pattern of DNA bands from an individual's genome. So here we have an example of a genetic fingerprint produced by the process of gel electrophoresis, which we will get onto in a sec. So by producing specific patterns, we can look at how closely related individuals are, and we can compare, for example, the DNA band profile from a suspect of a crime to the DNA profile from a crime scene, for example. Genetic fingerprinting uses pieces of DNA called VNTRs, which stands for variable number tandem repeats. So they are pieces of DNA that repeat themselves, and they have variable numbers amongst the individuals. So VNTRs, or variable number tandem repeats, are short repeating sequences of DNA, and they take place in non-coding regions or introns. Remembering that non-coding regions are regions of DNA that do not code for amino acids. In every individual, VNTRs vary in length and in the number of repeats, so the probability of two individuals having the same VNTRs is very low. Because of this, VNTRs are very useful in genetic fingerprinting. Because VNTRs differ a lot compared with different people, so we can identify people, so criminals, more easily. So here we have an example of a VNTR, so the CAG repeats. CAG codes for the amino acid glutamine, I think. So here we have individual 1 on the top here, which has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 repeats. And on the bottom here we have individual 2, which has 1, 2, 3, 4, 5, 6, 7, 8, 9 repeats. So different individuals have different number of repeats and lengths of VNTRs, so we can identify individuals very easily using genetic fingerprinting of VNTRs. So how is genetic or DNA fingerprinting carried out? Well, first the DNA needs to be extracted. So the extraction of the DNA of interest, and amplification by PCR, or the polymerase chain reaction. If you are not confident with the process of PCR, check out my video about recombinant DNA technology in the corner. The next step is that we need to digest the DNA to get the VNTRs that we want, so the genes of interest. To do this, we use specific restriction endonucleases that cut the DNA into DNA fragments, so the VNTRs are left behind. Then DNA fragments, or the VNTRs, are separated by gel electrophoresis. So here is an image of a gel electrophoresis, which is carried out most of the time on a gel containing the sugar agarose. So if I just get my pen tool out, the DNA fragments are inserted at the top of the plate. Then an electrical current is switched on and the DNA fragments move towards the negative electrode. This is because DNA has Actually, that is a typo, it should read positive electrode. So plus VE to denote positive electrode. So the DNA fragments, or the so the VNTRs, move towards the positive electrode because DNA is negatively charged due to the phosphate groups. So the DNA fragments move along the gel. However, smaller fragments, so those with less DNA-based sequences, move further down the gel as they are lighter, and larger fragments move a shorter distance. So larger fragments are found near the tops of the gels, and smaller fragments are settle near the bottom. And these different sized fragments are separated into bands. So if we have a band that is thicker, so this band here, that means that there's a higher amount of DNA that has a specific size. So bands are formed. So what happens next? The VNTRs, so your fragments moved by gel electrophoresis, are hybridized. So if you haven't got familiar with that, check out my video about DNA probes, are specific complementary base sequences with DNA probes. So DNA probes are short sequences of DNA that are complementary to the target sequence. So in this case, the target sequence are our VNTRs of interest. Then the gel is developed. So this means that the gel can be dried and then placed on an X-ray film, for example. So the pattern of bands can be visualized, so can be seen, by placing the gel on an X-ray film, as the probes can emit radiation, because probes often contain a radioactive tag. This reveals the positions of the bands, and then therefore we can know the genetic profile of an individual. So here we have a gel electrophoresis that has been developed onto an X-ray film. So here you can quite easily see the bands of DNA fragments here. So how can we use these gel electrophoresis for DNA profiling? We can determine genetic relationships by looking at how similar the bonding patterns are. So if the bonding patterns are more similar, the more closely related the individuals are. We can use DNA profiling for forensics, so comparing the DNA profile of a suspect with those found at the crime scene. So here we have an example of this. So here we have the bands of the crime scene sample, and here we have 3 suspects. The main suspect here is B, because as you can see, the bands are the exact same in the crime scene sample and with suspect B, as the bands are on the same level, which means that the fragments or the VNTRs are the same size. However, with suspects A and C, different bands are shown. So this thick band here in suspect A isn't present in the crime scene sample, for example. Also, genetic profiling or DNA profiling using electrophoresis can be used in medical diagnosis, so to identify individuals at risk of developing particular diseases, for example, Huntington's disease. As Huntington's disease is what we call a polyglutamine disorder. So if an individual has a too large amount of CAG repeats, like we said earlier, they have a high risk of developing Huntington's disease. So this demonstrates how VNTRs are very important in gel electrophoresis profiling. Also, profiling is used in plant and animal breeding, because this is used to prevent inbreeding by not breeding individuals with similar banding patterns. Inbreeding would be very, very disastrous, as it would lead to a lower genetic diversity and a higher prevalence of genetic disorders, as closely related individuals are more likely to carry recessive alleles for a particular disease. Right, so that is it for the content, and now I'm going to take you through some exam questions. Right, so let's read this one. Huntington's disease is a genetic condition that leads to loss in brain function. The gene involved contains a section of DNA with many repeats as a base sequence CAG, so like we mentioned earlier in the video. The number of these repeats determines whether or not an allele of this gene will cause Huntington's disease. So an allele with 40 or more CAG repeats will cause Huntington's disease. An allele with 36 to 39 CAG repeats may cause Huntington's disease. And an allele with fewer than 36 CAG repeats will not cause Huntington's disease. The graph shows the age at which a sample of patients with Huntington's disease first develop symptoms and the number of CAG repeats in the allele causing Huntington's disease in each patient. So here on this is sometimes what we call a dot plot or dot graph. So on the x-axis we have the number of CAG repeats. On the y-axis we have the age at which patients with Huntington's disease first develop symptoms in years. So the question says, people can be tested to see whether they have an allele for this gene with more than 36 CAG repeats. So we can highlight this. Some doctors suggest that the results can be used to predict the age at which someone will develop Huntington's disease. Use the information in the graph to evaluate this suggestion. Right, so this is what I've suggested. Also, note that the question says evaluate, so you need to make references to whether the results overlap with each other. So this is what I've written. The graph shows a negative correlation. So this means that the higher the number of CAG repeats, the lower age at which Huntington's disease first develops, as you can see by the pattern of the graph, which is generally going down. However, there is overlap at some points, especially here there is a lot of overlap. This suggests that other factors may be involved at the age of onset. So for example, your diet, for example, or something other than CAG repeats. So if we look at the mark scheme, mark point 1, negative correlation, if we put. Also here it accepts a description for negative correlation, so you could write that the higher the number of CAG repeats, the lower the age of onset. Neutral correlation, so you might not get a mark if you write correlation, as the correlation is specifically negative. Also, it says reject positive correlation, as that is completely wrong. It is a negative correlation. As it says reject also, if you put any reference to a positive correlation, then you do not get any marks at all for the question, no matter if any of these marking points here are right or not. So the second marking point that you could have written is that there is a wide range. We didn't put that, but that doesn't matter as there are other points to look at. Mark point 3, overlap. We wrote about overlap, so we would get a mark. And mark point 4, the graph suggests that other factors may be involved in the age of onset. We wrote that, so we would get all 3 marks for the question. Noting that the mark scheme says 3 marks, so if you put all 4 of these marking points, you do not get 4 marks, you can only get a maximum of 3 marks. Also here it says, for mark points 2 and 3, accept the use of figures from the graph, and for mark points 2 and 3 can refer to the age of onset or number of CAG repeats. So you could put about those 2 things. So you can use the figures themselves if you want. However, it says ignore references to methodology, because it doesn't really mention that about what methods they use to gather the number of CAG repeats in the question. So if we move on, Huntington's disease is always fatal. Despite this, the allele is passed on in human populations. Use the information in the graph to suggest why. Right, so as you can see with the general trend in the graph, there is a lot of people that have the age of onset at a higher age, so in their 30s or 40s. Now, generally at these kinds of ages, so older ages, people will have already had children before the age of onset. So this means that the allele is passed on to them. So this is what I've written. Individuals have already passed on the allele before symptoms occur, as a lot of Huntington's disease cases occur in your mid to later life, after you have already had children, so after you have already passed on the allele. So if you look at the mark scheme, mark point 1, you can either put these 2 points together, So the age of onset can be high or symptoms appear later in life. They accept gene for allele as well, although it prefers if you put allele. So individuals have already had children / the allele has been passed on. Or you can write these two marking points together. So individuals have passed on the allele or have already had children before symptoms occur. We put these two marking points here, so we would get all 2 marks for the question. Note how it says or, so you need to put either mark point 1 and mark point 2 together, or mark point 3 and 4 together. You can't put 1 and 3, or 2 and 4, or 2 and 3 together, for example. And if you put more than 2 of these points, you do not get more than 2 marks, as the mark scheme here says 2 max. So let's move on. Scientists took DNA samples from 3 people, J, K, and L. They used polymerase chain reaction, PCR, to produce many copies of the piece of DNA containing the CAG repeats obtained from each person. They separated the DNA fragments by gel electrophoresis. A radioactively labeled probe was then used to detect the fragments. The diagram shows the appearance of part of the gel, so it's not the whole thing, after an X-ray was taken. The bands show the DNA fragments that contain the CAG repeats. So here we have our gel here with the direction of movement of the fragments and the wells, which are the areas on the gel where the DNA sample is injected into. So if we look at the first part of the question, only one of these people tested positive for Huntington's disease. Which person was this? So if we look at each of the samples, with L we only have 1 band, and with K we have 2, and J we have 2. So we can say that K is the person that tested positive because K has a band which has the largest size of fragments as the band is near the top. So this suggests that the CAG repeats is larger. So the fragment will be larger and therefore not move as long as a distance as the other bands. So my explanation is that they have the band that moved the shortest distance, so the largest number of CAG repeats. So obviously they will test positive for the disease, as the more CAG repeats you have, the more likely you are to have Huntington's disease. So if we look at the mark scheme, mark point 1 is for stating that it is person K, and mark point 2, as it has the highest band or has a band that travelled the shortest distance, we wrote this point here, so we would get all 2 marks for the question. Or for this point, you could put that so has the largest fragment, or so the largest CAG repeats. So you must write 2 points to get the second mark. So you must correctly link the distance moved and the fragment size. We wrote that the band travelled the shortest distance, so that means that the person K has the largest number of CAG repeats, so we correctly linked the 2 factors, so we would get all 2 marks for the question. So let's move on to the next part of the question. The diagram only shows part of the gel. Suggest how the scientists found the number of CAG repeats in the bands shown on the gel. So as it is a suggest question, we need to come up with our own ideas, but we don't need to go into a lot of detail as this question is only worth 1 mark. So this is what I've put. They could use CAG repeats of known length at the same time. So if we put CAG repeats in that have a lower length, so under 35 or 36, which will indicate non-Huntington's disease, then we can compare that to our individuals in our experiment. So if they have a higher number than our known length CAG repeats, we can evaluate whether they will develop Huntington's disease. And we do this at the same time to allow comparisons as well. So if we look at the mark scheme, run fragments of known length or known CAG repeats at the same time. So we would get the mark for the question. Also, it accepts references to a DNA ladder or DNA markers. However, do not accept DNA sequencing, as gel electrophoresis does not sequence DNA and measures the size of DNA fragments. So let's move on to the next question. 2 bands are usually seen for each person tested. Suggest why only 1 band was seen for person L. Well, if only 1 band was in person L, this suggests that only one side of the DNA fragment was produced, suggesting that person L is homozygous. So I've written that their CAG fragments are the same length or the same size. Obviously, I do not need to go into more detail as this question is only worth 1 mark. So if we look at the mark scheme, person L is homozygous, or you can write the CAG fragments are the same length or the same size or the same mass. We wrote that the CAG fragments are the same length, so we would get the mark for the question. Also, it says here, except small fragment has run off the gel or travelled further, which could happen if you haven't done your experiment right. So let's move on. Right, so that is it for this video. Thank you very much for watching. If you have any questions, please leave them in the comments. I'll be more than happy to answer them, and I'll see you later.